So I'm now very, very happy to introduce Tanja Slotte, professor at in ecological genomics in Stockholm, and so she's doing a very, very nice work on evolutionary transition, in particular from outcrossing to selfing, and she will talk now on the evolution of Distyly in Linum, very nice work.
Right. So first of all, before I start, I just want to thank the organizers for putting together this very nice meeting and inviting me here to talk about our work on the genetic architecture and evolution of the Distyly supergene in Linum. And so, as many of us already know, supergenes can allow the maintenance of favorable combinations of traits due to suppressed recombination. And as we've heard in the previous talk, they occur in many different systems. So for instance, in fire ants, we're also going to hear about supergenes that govern mimicry in butterflies. But today I want to focus on a supergene that governs Distyly. And Distyly is a polymorphic trait that has long interested many generations of biologists. So in Distyly species, individuals have 1 out of 2 different types of floral morphs that differ reciprocally in the placement of their male and female reproductive organs, their anthers and their stigmas. So pin or long-styled morphs have the stigma in a high position in the flower and the anthers in a low position in the flower, whereas thrum or short-styled morphs have the stigma in a low position in the flower and the anthers in a high position in the flower. And in addition to these morphological differences, there's usually also physiological incompatibility, which means that only crosses between individuals of different morphs and not between individuals with the same morph can give rise to offspring. And in natural populations, there's usually approximately equal numbers of pin and thrum individuals. Now, Distyly is an iconic example of phenotypic convergence. It has arisen independently many different times in flowering plant evolution. So here on the left-hand side, I'm just showing you 2 examples with pin and thrum flowers of Primula and Linum, and these species are diverged by about 100 million years and have independently evolved Distyly. So this suggests that selection for Distyly can be quite strong. What then is the adaptive significance of Distyly? Well, currently it's accepted that Distyly is adaptive because it enhances efficient pollen transfer between morphs and allows avoidance of sexual interference. So as you can see in the schematic drawing, pollinators pick up pollen at this location here in the pin morph and deposit it here on the stigma of the thrum morph, and vice versa here in the other direction. And of course, the self-incompatibility also allows for inbreeding avoidance. And so it was actually Darwin who first proposed that the adaptive significance of Distyly was efficient pollen transfer, and he considered that one of his most important discoveries. Early geneticists were also really fascinated by Distyly, and through crosses they showed that it was inherited as if it was governed by a single Mendelian locus, with 1 dominant allele that was found only in thrums, and 1 recessive allele for which pins were homozygous. And they did this first in Primula, but later on also in other Distylous systems, and in most of them it looks like this. And so to explain how you could have these multiple morphological and physiological traits being co-inherited always in specific combinations, they turned to the supergene model. And they hypothesized that there were multiple genes with 1 dominant and 1 recessive allele that were locked together in a region of the genome without recombination. And I've just schematically depicted that here. And so in Primula, Ernst even went so far as to delimit that number to 3 genes. And so if you look at this inheritance model, you can see that you might expect the dominant haplotype to start accumulating deleterious mutations, because it's morph limited. It's found only in half of the individuals in the population, so it has a reduced effective population size. It's not expected to undergo recombination, and if this classic model is correct, then it should also have masking of recessive deleterious mutations. So some similarities or parallels can be seen between this kind of system and XY sex chromosome systems. But until recently, it wasn't possible to test this hypothesis because no single Distyly supergene had actually ever been sequenced. This changed in 2016 when the first sequence of the Distyly supergene was presented for Primula vulgaris. And to everybody's surprise, what they found was that this Distyly supergene was not actually heterozygous in thrums, but it was hemizygous. So it harbored a thrum-specific region that was simply lacking from the recessive allele. And later on, a similar genetic architecture has been shown to govern Distyly in Ternera, which has independently evolved Distyly. And based on that, it has then been suggested that perhaps hemizygosity in thrums is actually a common feature of independently evolved Distyly S loci. It would be important to understand if that's the case, because hemizygosity at the Distyly supergene will affect the evolutionary trajectories of the two haplotypes, and it will also modify expectations on the genetic basis of breakdown of supergenes, which has happened frequently in the case of Distyly. But to determine whether this is actually a common occurrence or a general finding, we need to study the genetic architecture of Distyly supergenes in more different systems with independently evolved Distyly. And so I have chosen to do that in Linum, or wild flaxseed species, which is actually a classic system for studying Distyly. Darwin described Distyly in Linum in 1863. And in this system, several independent gains and losses of Distyly have been inferred, suggesting that this could be a good model system to study the evolution and loss of supergenes that govern Distyly. There's also a wide diversity of species, many of which are found in the Mediterranean region. And finally, Linum and Primula have convergently evolved Distyly. And so in the ERC project Supergene, we are sequencing high-quality genomes of 3 pairs of Linum species with and without Distyly. We're also sequencing a couple more with funding from the Swedish Research Council, in order to use these genomes as a framework to be able to test hypotheses on supergene origins and evolution. And we're also interested in understanding the genetic basis and population genomic consequences of the breakdown of Distyly. And here I'm going to focus on one of these species, Linum tenue, which I'll introduce a bit more. So Linum tenue is a Distyly species that occurs in these kind of dry meadows in southern Spain and in North Africa. It's distylous. This is what the flowers look like in the field, and this is what they look like if you take off the petals and sepals. It's pollinated by syrphid flies and bee flies. And this is a diploid annual species with a short generation time, making it very suitable for genetic work. So we were really happy to go in the field and find lots of seeds of this. And so we grew those to get DNA and make high-quality genome assemblies. So we opted for high-coverage pack biosequencing as a basis, and then did scaffolding using high-C and polishing using various forms of Illumina sequencing. And we also generated a linkage map to anchor our scaffolds to pseudochromosomes and annotated these genomes using RNA-seq data from 5 different tissues. And the reason why, of course, we're interested in getting as high a quality genome as possible is because supergenes are often highly repetitive. So we want to have a good coverage also in those regions. And so in Linum tenue, which was the species we started with, we sequenced both morphs. And the assemblies that we retrieved using this approach were highly contiguous and complete. They had 10 pseudochromosomes, which is the haploid chromosome number of this species. The genome size is approximately the same as what we measure using flow cytometry. And N50 and Basco scores also indicate high contiguity and completeness. So now we had a genomic framework that we can use to find the supergene and study its evolution. And this is exactly what my PhD student, Juanita Gutierrez Valentia, did. She used samples of pin and thrums from natural populations, resequenced them, and did the simple GWAS of floral morph, and found a single peak on chromosome 10. That showed a strongly significant association between SNP genotype and floral morph. And if we zoom into this region, it's still highly significant. It's still one region also. And then we looked into structural variation in this region because we expected that this might be something that could occur given the results from Primula and Turner. And in this region, we do see actually a really strong coverage drop in pin individuals compared to thrum individuals. And also thrum individuals have about half the coverage in this region compared to flanking regions. When we compare our phased haplotypes from the thrum and pin genomes, we see that there's a region here that is only found in one haplotype in the thrum and not in the pin individuals, and we have verified this using link tree assemblies from multiple more individuals. And so what it looks like is that we do have a situation where thrums are hemizygous for this part of the S locus, and pins are simply missing that region. Is that all, or could recombination be suppressed in a broader region? Well, the association between morph and genotype does not really extend very far outside of this 260 kb indel, only about 15-20 kb on each side. And if we look at FST between pins and thrums, we see elevated FST between them, but only right next to the indel, not stretching far outside as you would expect if there was a bigger region with suppressed recombination. And then we used our genetic map to further see whether there was any evidence for broad-scale recombination suppression around this structural variation, and we did not find that. So the supergene is actually located in a region that has fairly high recombination rates, and also if we look at linkage disequilibrium decay in natural populations, we don't see evidence for extended linkage disequilibrium far outside of this supergene. So what this suggests is that it could be that the structural variation at this supergene is indeed instrumental for recombination suppression at the supergene. And so these results show remarkable convergence in the genetic architecture of the Distyly supergene between Primula and Linum. So in Primula, if you recall, they had identified an approximately 280 or 260, depending on which species you look at, thrum-specific region on the dominant S haplotype. And in Linum tenue, we have identified an approximately 260 kb thrum-specific region, also on the dominant S haplotype. So this suggests that if this holds more generally, the classic model for the inheritance of Distyly should indeed be revised. What are then the consequences of hemizygosity for the evolution of supergene haplotypes? Emma Burden, who is also here today, did some simulations in SLIM to show that we do expect a slower accumulation of recessive deleterious mutations in a dominant S haplotype when you have an indel genetic architecture compared to if you would have had an inversion genetic architecture. And so what we find when we investigate patterns of molecular evolution at the Linum tenue Distyly supergene is that we don't find any significant elevation of non-synonymous to synonymous polymorphism in the S-linked region compared to neighboring regions. We're going to look into divergence data, but we don't quite have that yet. But what we do find is elevated fraction of repeats in the S-linked region compared to neighboring regions and also compared to the chromosome 10 as a whole. And again, these patterns are remarkably similar to what has previously been observed in Primula. So both repeat enrichment and weak signatures of genic decay have recently been documented at the Distyly supergene in Primula, which has a similar genetic architecture. So there's both convergence at the level of genetic architecture and apparently also when it comes to molecular evolution. What are then the genes within this supergene region? Can we identify any candidate genes for Distyly? Yes, we can. So overall, this supergene harbors just 9 protein-coding genes with diverse functions, excluding some genes that are transposon-derived. Most of them are complete, with the exception So most of them are found only on the dominant haplotype. There's one gene that is also found in the recessive haplotype, and then there is another one where the recessive haplotype copy is truncated. The most striking candidate gene that we have in this region is this gene here, LTTSS1. And this is a very strong candidate gene for style length polymorphism for several reasons. So first of all, this gene is homologous to a previously identified gene called TRUM style-specific 1 that was identified as a TRUM-specific gene with style-specific expression in this species, Linum grandiflorum. In our own data, so we generated expression data from a number of different floral organs and leaves and whole buds. We can see that also in Linum tenue, this gene LTTSS1 is TRUM-specific, and it's only detectably expressed in buds and pistils. We also know that the shorter styles of TRUM individuals compared to PIN individuals is likely due to a reduction in style cell length in TRUMs. Now, interestingly, LTTSS1 has a homologue in Arabidopsis, which actually results in reduced cell and organ length when you overexpress that gene. And when we investigate differential expression between morphs, we see that genes that promote cell expansion are downregulated in Linum tenue TRUM pistils, as you might expect if the TSS1 gene is resulting in downregulation of those pathways. How old is then this supergene? Well, as we've heard, this is a question that is not always so easy to answer, and I suspect that we will need to come back to this probably many times with more data. If one were to guess at this stage, I can say that what we do see is that there's shared presence-absence polymorphism for TSS1 between Linum tenue, which is here in the latest Linum phylogeny, and Linum grandiflorum, which is here. And so if this is ancestral, that would mean that this polymorphism could have been present since the split of these species about 30 to 40 million years ago. We'll see. I definitely think we need to do detailed follow-up analysis on this, taking into account the possibility of introgression. And this is something that is currently ongoing based on genome assemblies that we have produced for several additional Linum species. How did then the gene presence-absence come to be? So it's been suggested that stepwise, that segmental duplications could be important in generating these types of supergenes with indel variation. If that were the case, then we might expect to find paralogs to the S-linked genes in one or just a few genomic regions of Linum tenue. We don't actually find that. We find paralogs of S-linked genes all over the genome of Linum tenue, and matches to S-locus genes were also on separate chromosomes in outgroups, suggesting that it's not the case that there was a single stepwise duplication resulting in this presence-absence variation. So our results are again very similar to those in Primula and suggest stepwise assembly of the genes set at the S-locus. This doesn't mean that that means stepwise assembly of the supergene. So the genes that can be assembled first and the polymorphism evolve after. So to summarize, our results suggest striking convergence with respect to hemizygosity in TRUMs, patterns of molecular evolution, and assembly of gene sets at the Distyly supergenes of Linum and Primula. And one possibility for why we see this repeatedly could be that dominant expression of S-linked genes facilitated establishment of mutations contributing to first steps of Distyly evolution. The presence of TSS1 as an S-linked presence-absence polymorphism in tenue and grandiflorum suggests that this polymorphism could be quite old, but we need to revisit this using more data. And finally, this work improves the foundation to understand Distyly, supergene evolution, and the genetic basis of Darwin's different forms of flowers. And with that, I want to thank everybody who contributed to the work, especially Juanita Gutierrez Valencia, Marco Fracassetti, and Emma Burden, but also collaborators in Spain and the UK, and bioinformatics and sequencing platforms in Sweden, and funding, of course. And I also have a postdoc available. Thank you.